All right, well, welcome back. Last time we finished up muscles of the shoulder girdle. So we're going to insert one of the textbook chapters here, and we're going to talk about muscles and muscle tissue here in part so we have a little space so that you can study up for the lab quiz that's coming up over the shoulder girdle. And then we'll kick back into orthopedics mode, if you will, and we'll get back into the bones and muscles of the forearm. So, as far as muscles and muscle tissue goes, so most of this follows pretty closely to the textbook, as you'll see from my slideshow, but there are several things that I've added that are not in the textbook. So in terms of types of muscle tissue, I should probably back up a step and tell you that when we talk about a tissue, that is a group of cells of the same type working together. So in the case of skeletal muscle tissue, you've got a bunch of skeletal muscle cells that are all working together, and so then they form a tissue. And an organ, which is related to a tissue, an organ is multiple types of tissue working together. So in the case of an actual skeletal muscle, so something like your biceps brachii, that is an organ because it includes not only skeletal muscle cells, but it also includes connective tissue cells. It also includes arteries, veins, nerves. And so because of those different types of tissue that are all working together, then that becomes an organ. So in terms of muscle tissue, what I'm saying by that, or what I mean by that, is multiples of the same type of cell working together. So as far as types of muscle tissue then, you've got skeletal muscle, cardiac muscle, and smooth muscle. So far in this class, we've only talked about skeletal muscle. So skeletal muscle, some important characteristics of it. The first is that it's voluntary, right? So all those muscles that we talked about, whether it's biceps brachii or pectoralis major or infraspinatus, all those muscles, you can voluntarily contract them. So you can think about, I want to lift my arm, and so then you're voluntarily engaging biceps as a flexor at the glenohumeral joint. So in addition to being voluntary muscle, skeletal muscle has some other important qualities, and you can see these kind of in the lower right there. So among them is that skeletal muscle is sometimes also referred to as striated muscle, and so striated means striped. And you can't see it on the picture down here unless you really zoom in, kind of. So there's some sort of tiger stripes to the muscle here, right? So when you look at skeletal muscle cells under a microscope, they kind of have a striped appearance. And the reason for that appearance is there are certain areas within the cell of overlap or lack thereof between the contractile proteins. So the contractile proteins in muscle cells are actin and myosin. And we'll talk about them in more detail later. But where you've got areas of overlap between actin and myosin, that appears as a dark stripe. And then where there are areas where there aren't overlap, where it's just actin or it's just myosin, that appears lighter under the microscope. And so that's why you've got that light-dark alternating appearance to them. So that's striated, or they are striated, and so is cardiac muscle. So other things about skeletal muscle cells, so they're voluntary, they're striated, they're also multinucleated. So what does that mean? Well, so you're aware from your high school biology classes or the biology class you had prior to this one that cells generally have a nucleus to them, right? And so the nucleus of a cell, what it does is it contains the DNA. And so DNA, one of the ways to think about that is that it is the instructions for building proteins. So actin is a protein, myosin is a protein. Again, those are the two contractile proteins in a muscle cell. But we also have proteins embedded in the cell's membrane, and then proteins in various other areas throughout the cells. So in order to have those proteins, we have to manufacture them, we have to make them. And so the instructions for making them are contained inside of the nucleus. And so if a cell is multinucleated, if it has multiple nuclei, what we can infer then is that's a cell that makes a lot of protein. So in the case of skeletal muscles, they have to be able to make additional proteins, right? So to be able to repair themselves. So for example, if you undertake a strength training program, if you go lift weights and you do that over the course of several weeks, eventually your muscles are going to get bigger. And the way that they're going to do that, one of the ways, is they're going to add additional actin and myosin. And so you're going to make new proteins. And so in order to do that, again, you need the instructions for actin and myosin. And so those instructions are contained in the DNA in the nucleus. So we've got multiple nuclei so that we have lots of different sets of instructions to teach us or to tell the ribosomes. The ribosomes are the organelle that is going to make those proteins. So the instructions are used by the DNA, being the instructions, is used by the ribosomes to make those proteins. So skeletal muscle cells are also multinucleated. They make a lot of protein. No surprise there. So those are the three big things I want you to know about skeletal muscle. It's voluntary, it's striated, which means it's striped, and then it's multinucleated. Cardiac muscle, just a quick bit about it because we're going to talk about it a lot in PEPROF 202. But for now, so anything cardiac deals with the heart. And so cardiac muscle is the muscle in the walls of the heart. It's similar to skeletal muscle in that it's striated. It can have more than one nucleus, but typically only has one. So 1 to 2 nuclei. And then, in addition to that, actually those are the two big similarities there. So striated and potentially has more than one nucleus. So those are the similarities. But cardiac muscle, of course, is not voluntary. So your heart beats on its own. Now we can affect that if you're, scared or excited or whatever. You can increase your heart rate, or you can decrease your heart rate. By calming down, and a number of different exercises you can do. So we have some influence over it, but it's not voluntary in the same way that skeletal muscle is voluntary. So cardiac muscle then, involuntary muscle. Some other differences. So in the case of a skeletal muscle cell, and I should clarify here, when I'm saying skeletal muscle cell and skeletal muscle fiber, I mean the same thing. So muscle cell, muscle fiber, as far as skeletal muscle goes, those are the same thing. In the case of skeletal muscle cells, the cell runs all the way from one tendon to the other. So they're really long cells or really long fibers, depending upon which muscle we're talking about. So if you're talking about the sartorius, which is one of the muscles in your thigh, it's the longest muscle in the body, those cells can be 18 inches long, right? So those are really long cells. In some of the little muscles of the eye, they're only a few millimeters. But in the case of skeletal muscle cells, again, those are going to run from one tendon to the other, so they can be really long cells. Cardiac muscle cells are much shorter, and they don't run because there aren't tendons per se in the heart, right? There is connective tissue, but there aren't tendons per se. So cardiac muscle cells actually run into one another, versus skeletal muscle cells run in parallel to each other. And so you can actually see that on this drawing down here. So here's one skeletal muscle cell right here, and then running right alongside it, here's another one. Right, so they don't intersect with each other. They just run from one tendon over here to the other. As opposed to in your cardiac muscle cells, here's one cell, and it runs directly into another cell. And then that one branches a little bit and then runs into, so here's where that cell ends, and then it runs into this cell or another cell right here. So cardiac muscle cells, rather than running in parallel, are actually branched. And then, again, they connect to each other. And the reason that that's important is that allows them to distribute the electrical signal, causing the heart to contract really evenly, because we need the heart to contract in an orderly fashion. So in the case of the ventricles, which are these lower chambers down here, we need the heart to contract in a bottom-up sort of a fashion to wring blood from the left ventricle here out into your aorta, the red thing here, and then it goes out to the rest of the body. So if you've got a disorder in terms of the firing of those cardiac muscle cells, well, obviously that's a problem. So if you've got some cells, up here, higher up in the wall of the left ventricle contracting, and then one down here, and then one up here, that's fibrillation. That's just kind of a shaking or generalized contraction. And then that's not productive. That's not going to pump blood. So in order to minimize the risk of that as much as possible, those cardiac muscle cells run into each other to evenly distribute the electrical signal that stimulates them to contract. Cardiac muscle cells, a couple other fun facts about them. They're very aerobic. They're really, really good at using oxygen. So they're able to use sources of fuels, if you will, that other muscle cells are not as good at using. So for example, they can use lactate, which can be a byproduct of muscle contraction, of skeletal muscle contraction. They can use that as fuel because they're really good at using oxygen to reproduce the energy molecule, which is ATP. And then the last one there is smooth muscle. So smooth muscle is called smooth because it's not obviously striated. It's not striped when you look at it under a microscope. And so you can see that here, no stripes. Smooth muscle, the thing I want you to know about it is it's found around the walls of your hollow organs. So what does that mean? It's found around your esophagus, which is when you swallow food and it goes down into your stomach. That's your esophagus. So it helps wring the food from your mouth down into your stomach. The stomach is surrounded by smooth muscle that helps it contract and pulverize the food. And then your intestines are also surrounded by smooth muscle that help propel the things you eat through them so you can absorb the nutrients you need and then expel the things that are left that you can't use. So smooth muscle then, again, surrounds your hollow organs. So things like the esophagus, the stomach, and your intestines. All right, so back to skeletal muscle specifically. So in terms of characteristics of muscle tissue, so you can see the first one there is excitability. And so excitability means that it can respond to stimuli. So muscle cells receive input from the brain via nerves, and the brain sends an electrical signal. When it wants a muscle to contract, it will generate an electrical signal that then goes down your spinal cord, out the nerve, and to the muscle cell. And so that electrical signal causes the muscle cell to respond. So it responds to that stimulus, and it responds by contracting. So excitability means that muscles respond to a stimulus, and then contractility is that muscles shorten. So, and an important thing here is that muscles can only forcefully shorten. So muscles can't forcefully lengthen. Usually gravity does that, or antagonistic muscles cause a lengthening of a muscle. So muscles are contractile, meaning that they shorten. So they pull one tendon toward the other. So for example, whenever you flex your elbow, which is to bend your elbow, you're going to pull the insertion of biceps brachii at the tuberosity of the radius toward the origins. So again, the coracoid process and the supraglenoid tubercle. So when a muscle contracts, it typically pulls the insertion toward the origin most of the time. So that's contractility. It's the shortening of the tissue. So think back to what we talked about in terms of shoulder stabilizers. So remember that for the glenohumeral joint, I included on the slide that there were static stabilizers, which are non-contractile, and dynamic stabilizers, which are contractile. So remember that the static stabilizers include the ligaments, which connect bones to bones, the joint capsules, which also connect bones to bones, and then the cartilage within the joint. So in the case of the shoulder, the glenoid labrum. So none of those structures are able to contract, none of them shorten. All of them can lengthen a little bit. You can stretch a ligament out, and then it'll actually return back to its original shape, but it won't get any shorter than its original shape. Okay, so that is another property we'll talk about here in a second. So those tissues then, the non-contractile tissues, those are your static stabilizers. Your dynamic stabilizers, the moving stabilizers, well, those are the muscles of the rotator cuff and also biceps brachii's long head. So those are going to keep the humeral head in connection with the glenoid throughout the range of motion. So those are your dynamic stabilizers. They're your contractile tissue. So muscles are able to shorten. And then a couple other terms on here. You've got extensibility. So if something is extensible, that means it's capable of being stretched. So muscles can obviously be stretched. And then same thing for those static things we talked about, the static structures we talked about. So your ligaments and your joint capsules can also be stretched. And then elasticity is the capacity to change shape and return back to the tissue's original configuration. And so I worded that way because typically we think of elasticity, if we think of something that's elastic, we think of something like a rubber band. So with a rubber band, you can stretch it out, and then it returns back to its original length whenever you let go, right? And so muscles do that. Muscles are capable of being stretched and returning back to their original length, so that's elasticity. But elasticity is also the ability to be compressed and return back to the tissue's original shape. So we also see elasticity in your cartilage because it can be compressed and then bounce back. And we also see it in the bone as well. So I just wanted you to be aware that elasticity is not only the ability to return to the tissue's original shape from being stretched, but also the capacity to return to original shape from being compressed. So muscles are not the only tissues that are elastic. So what this lady on the bottom right is demonstrating is essentially all of those characteristics of muscle, right? So she is voluntarily contracting her quads, her hamstrings, and her glute muscles in order to extend her hips and extend her knees and to propel herself onto the top of this box. So she's getting a signal coming down from her brain, causing those muscles to contract, right? So they're responding to that stimulus from the brain. So you got excitability and contractility there. And then an important thing she's doing on the left, this is called a counter movement. And so in a counter movement, what you do, like if you want to jump on a box like she is, you're probably not just going to jump from standing. You're probably going to drop into a really quick squat first and then jump up. And so that's the counter movement. That's a quick stretch of a muscle and then contraction shortly thereafter. So what we're getting there is she's showing the extensibility, so the ability to lengthen her quad muscles in particular, and then their elastic characteristic that they want to return back to their original shape. So as she stretches the quads, again, they're going to lengthen, but then they want to return back to their original shape. So that connective tissue around the quads is going to, I'm going to say retract, to try to return to its original shape. And that is going to augment or assist the contractile component of the muscle. And so that actually helps you jump a little bit higher. So if you stretch a muscle first, because of its elastic quality or characteristic, that is going to assist the contractile component. And so that helps you then jump higher or throw harder or those kinds of things. All right, so in terms of muscle functions, so there's some pretty obvious ones on there. So muscles obviously produce movement, so they help us walk or run or throw or do any of those other things. They move one bone around another or relative to another, and that then produces movement. In addition to that, so they help us maintain our posture. So you've got muscles in your neck, for example, or in your upper back that stabilize your spine, that extend your neck. And so if you extend the spine or if you extend the neck, that's to look straight up at the ceiling. And so you've got some neck extensors right now. If you're seated in a chair as I am and looking at the screen, you've got some neck extensors that are maintaining posture, that are stabilizing your head and keeping it in a nice neutral position so that you can look at the screen. In terms of body position, so muscles can contract and may not necessarily change a joint angle, but they help us maintain body position. So for example, if you're on ice, right? So if you're standing on ice, obviously that's going to be pretty slippery and that may tend to allow your legs to slide apart, and so that could cause you to fall. So you're going to get an isometric contraction, and we'll talk more about that later, but that's where you get a contraction of the muscles around a joint just to stabilize it. You're not trying to cause any movement, but you're just trying to stabilize that joint. So you try to stabilize your hips to keep your legs from sliding apart, which would then cause you to fall. So you can maintain body position, you can stabilize joints, and that's another important thing that muscles do. So obviously then these two are interrelated. In terms of joint stability, stronger muscles around a joint make for a more stable joint, make it less likely that you're going to move those two joint surfaces apart. So for example, since we've talked about the shoulder, again, it's not uncommon to have an unstable shoulder. So if somebody, I've talked a lot about throwing athletes, so that throwing motion tends to stretch out those glenohumeral ligaments. So particularly that middle glenohumeral ligament and then the anterior portion of the inferior glenohumeral ligament. Well, if those ligaments, if those static stabilizers are stretched out, then you're going to get extra motion between the humeral head and the glenoid. And so one of the ways that we can minimize that, because again, that could cause pain or dysfunction, one of the ways that we can minimize that is to do rotator cuff strengthening. So you can do resisted internal rotation, resisted external rotation, and some resisted abduction in different planes to try to strengthen up the muscles of the rotator cuff, because the stronger those muscles are, the more they're going to be able to pull the humeral head against the glenoid and sort of pick up the slack for those lax ligaments. So muscles play an important role in stabilizing joints. And then the last one on there is they generate heat as they contract. So muscles run on an energy currency. The actual molecule that provides the energy for muscles to contract is a molecule called adenosine triphosphate, or ATP for short. And I'm going to talk a lot about ATP. So ATP is the actual, again, it's the energy molecule. We split one of its bonds, and that the energy given off when we split that bond allows us to contract the muscles. And again, we'll talk about that in more detail in a minute, but also in future sessions. So when you break down that ATP molecule inside of the muscle cell, only about 25% of the energy given off actually drives muscle contraction, actually causes the muscle cell to shorten. The remaining approximately 75% is just given off as heat. And so when we contract muscles, we generate heat as sort of a byproduct of muscles not being 100% energy efficient. So, and the more energy you need, the more ATP you need, which you do, the more intense the exercise is, the more heat that you give off. So if you just go for a walk when it's cold outside, you're probably still going to be cold, unless you have enough layers on, because the energy requirements are pretty low. You're not running through very much ATP, so you're not giving off very much heat. But if you're outside sprinting, you're going to need a lot of energy to do that. You'll need a lot of ATP, and so the more ATP you break down, the more heat you give off, and so then that increases your core temperature, makes you hotter, right? And you also know that if you're cold, you involuntarily start to shiver. Well, what is shiver? Just involuntary, or shivering, it's involuntary muscle contractions. And so if we're just contracting the muscles but not actually producing any movement, we're burning through ATP to try to generate heat that way. So muscles also play an important role in helping us maintain our core temperature. They help keep us from getting too cold, so that we can continue all the cellular processes we need to in order to stay alive. All right, so let's talk some skeletal muscle anatomy. So here I'm going to toggle back and forth a little bit between the slideshow and the anatomy app, because the anatomy app actually has two really cool little models that we can use for our study of muscles. So you can see there, I'll read the slide to you for a second. The skeletal muscle is an organ made up of different tissues. So that was kind of some of the stuff I told you at the beginning. So the definition of a tissue, several of the same type of cell working together, and an organ then is multiple different types of tissues working together. So in the case of a muscle, like if we think about, again, biceps brachii, you've got nerves, blood supply, connective tissue, but then also skeletal muscle cells in there. So in terms of the nerve and blood supply, so each individual muscle cell or muscle fiber is going to have a connection to a nerve cell, which is called a neuron. So each muscle cell has a connection to the central nervous system. In addition to that, you're going to have blood vessels inside of the muscle and adjacent to or next to individual little muscle cells. We have an arterial supply. So again, or not again, I haven't told you this yet. So arteries bring oxygenated blood, so blood full of oxygen, to a tissue. And then veins bring deoxygenated blood, or blood with a lowered oxygen content, away from the tissue. So, and I'll actually click over here to my model, if it'll let me. There we go. All right, so we're going to look at the muscle model. So how you get there, this is the normal home screen. After you click on models, it goes to the skeleton here. So if you then go down here and click on models in the lower left, one of the things you'll see is a skeletal muscle. So that's the model that we're going to look at now. All right, so here's my generic skeletal muscle. So this whole thing is the muscle, okay? And then the stuff I was just talking about are the nerve and blood supply. So I'm going to remove this layer of connective tissue, and I'll talk about it in a second. Okay, so what you can see, these are individual little muscle cells. So there's one muscle cell, there's the one next to it, there's another one. And so this yellow thing, that's the nerve tissue. So that's going to provide the electrical signal that's going to cause these muscle cells to contract. And then you've got these red structures here. And so those are the arterial system. So they're bringing freshly oxygenated blood from the heart out to the muscle fibers. And then you've got this blue thing as a vein. And so they're going to return deoxygenated blood back to the heart, and then it'll go to the lungs to pick up more oxygen. So each individual little muscle cell is surrounded by capillaries. And so and the number of capillaries depends upon the type of muscle cell, but also can depend upon your training status. So if you're an endurance athlete, individual little muscle cells will have more capillaries around them. If you're not an endurance athlete, they tend to have a few less. So that is something that is variable, something that changes with response to, or in response to, training stimuli. So skeletal muscles then have nerve supply, causing them to contract. Because we'll talk about this a little bit more when we get to the nervous system. You also have nerves that run the other direction. You've got sensory nerves that let you know if the muscle is stretched or not. So that's part of the nerve supply as well. And again, blood supply. So I mentioned a minute ago, ATP, the energy molecule that actually drives muscle contraction. In order to make ATP, or remake ATP, we are going to need oxygen. So we need then that arterial blood to supply the muscle cell with oxygen, to deliver that to the muscle cell so that it can use that oxygen to help remake ATP, which gives us the fuel, if you will, for muscle contraction. And then there are, of course, byproducts of the energy production process, the process of making ATP. And one of those byproducts is carbon dioxide. And so the venous system is going to bring that blood with extra carbon dioxide, again, a waste product of energy production in the cell. It's going to bring that blood richer in carbon dioxide back to the heart and then back to the lungs, where we can expel that carbon dioxide. So the venous system helps the cell get rid of waste products. The arterial system brings oxygen to the cell so that it can remake ATP. All right, so we've also got connective tissue sheaths. So I'm actually going to go back to my original model here, click off of that. All right, so we'll zoom in a little bit. So there's 3 layers of connective tissue in a muscle. So the order of them from superficial, which remember that superficial means close to the skin surface, to deep, which means farther from the skin surface. So again, 3 layers of connective tissue. The most superficial layer is called the epimysium. And so I'm going to click on it so you can see the spelling. So the epimysium is a layer of connective tissue that wraps around the entire muscle. So it is a dense type of connective tissue, and the fibers are irregularly arranged, meaning that you've got some collagen fibers that run this way, some that run the opposite direction, some that run straight up and down, etc. So it's a really dense type of connective tissue. Sometimes it's referred to as fascia, and actually all 3 of these layers are sometimes collectively referred to as fascia, and that's okay. Just realize that fascia refers to the layers of connective tissue in and around a muscle. All right, so there's our epimysium, the most superficial of the 3 layers of connective tissue. And then, so again, the epimysium is going to surround the entire muscle. And then if we zoom in a little more, I'm going to move my muscle down some, we've got these bundles of muscle fibers. So this is a group of muscle fibers right here that I'm mousing over. So that's called a fascicle. And fascicle is spelled F-A-C-I-S-C-L-E. Be plain spelling bee. But fascicle will pop up in a second here when I click on the layer of connective tissue that surrounds it. So that's called the perimysium. So the perimysium is a layer of connective tissue that surrounds a fascicle, and a fascicle is a bundle of muscle cells. And in case you missed the spelling, it's over here all the way on the left. That's fascicles. So there's that. All right, so you got the perimysium surrounds a group of muscle cells. And then we've got, if we zoom in even more, there we go. So here I've got my individual little muscle cells right there. So where it says extrafusal muscle fiber, that's just your normal muscle fiber that we're going to spend our time talking about. And then each individual little muscle fiber has a layer of connective tissue around it, which is called the endomysium. And so the endomysium then is a layer of connective tissue that surrounds an individual little muscle cell. So 3 layers of connective tissue in a muscle. You've got the epimysium, surrounds the whole muscle. The perimysium surrounds a group of muscle cells or muscle fibers, which is called a fascicle. So the perimysium envelops or wraps around fascicles. And then the endomysium wraps around individual little muscle cells. So all 3 of those layers are going to converge together. So the endomysium runs into the perimysium, runs into the epimysium down at the end of the muscle. If I can zoom back out. Okay, and so this white stuff down here, that's the muscle's tendon. And so remember that tendons attach muscles to bones, and so you've got the belly of the muscle, which is all of the contractile part of it, and then this is called the myotendinous junction, so where all those layers of connective tissue fuse with the tendon. And so now down here, all you have is tendon, all you've got is collagen. So we'll talk about tendons in more detail later. And so if we go all the way down here, this would actually attach to the bone. All right, in terms of attachments, so on the slide, it says that attachments can be either direct or indirect. And so what that means is, I'm actually going to go back to the full body to show you, because I want to show you a specific muscle that's a pretty good example of having both types of attachments. So I'm going to zoom in on his posterior shoulder, and we'll add some muscles. Lots of layers of muscles. There we go. So let's go with infraspinatus. That's a pretty good example. And so is supra. That's fine. So where you've got a direct attachment, those 3 layers of connective tissue fuse directly with the bone. As opposed to if there's where the muscle cells don't, or the layers of connective tissue don't directly fuse to the bone, maybe there's a little bit of a gap there, that's an indirect attachment. So tendons are indirect attachments, and so you can see the indirect attachment over here, where infraspinatus inserts at the greater tubercle. And then you've got direct attachments over here along the spine of the scapula and then along the medial border. So where those layers of connective tissue fuse directly with the bone. All right, so that's those. And let me make sure I'm connected back to the internet. All right. Okay, sorry about that. Got my internet connection back. So covered all the stuff on this slide, so we'll move up one. All right, so let's talk about some terminology as it pertains to the individual little muscle cells or muscle fibers. So what you're looking at here, so this is an individual little muscle cell or muscle fiber. So the first term on there, the sarcolemma, that is the muscle cell's membrane. And so the sarcolemma on this cell depiction is right here. So that's the skeletal muscle cell's membrane. And then you've got the sarcoplasm. So sarcoplasm sounds a lot like cytoplasm. So remember, thinking back to what you know about generic cells, the cytoplasm is kind of that jelly-like material that all of the organelles float around inside of the cell. And so in the case of a muscle cell, the sarcoplasm is a muscle cell's cytoplasm. So it's that jelly-like material that everything floats around in. So here, this kind of yellowish stuff, that is this muscle cell's sarcoplasm. So that's the water or fluid component of the muscle cell. A couple other things on here. So we've got our glycosomes, or more specifically, glycogen. So what is glycogen? It is the intracellular, so inside of a cell, storage form of carbohydrates, and carbohydrates are sugars. So muscle cells, one of the things that they can use to make ATP is sugar. And so muscle cells then can store sugar in the form of glycogen inside of themselves, so in the sarcoplasm, so that sugar, glucose, is readily available as a fuel to remake ATP. And so you can see that here. So here's the little glycogen droplets in the sarcoplasm. There's also myoglobin. So myoglobin is not depicted on this drawing, nor is it on the anatomy app, but myoglobin has a relative. I don't know whether to call it a cousin or a sibling, but so inside of your red blood cells, you have a protein called hemoglobin. And so hemoglobin is the protein inside of red blood cells that actually transports oxygen. So myoglobin, again, is a relative of hemoglobin. And so myoglobin is a protein that's inside of the sarcoplasm, or that floats around in the sarcoplasm, that binds to oxygen, and it acts kind of like a shuttle bus for oxygen. So it's going to take oxygen from the surrounding capillaries, which are not pictured on this drawing, but it'll take oxygen from one of these surrounding capillaries. That oxygen will diffuse into the sarcoplasm, it'll bind with myoglobin, and then myoglobin is going to shuttle it over to where it needs to go, which is the mitochondria. So these little things are mitochondria, right there. And so hopefully you'll remember from your previous biology classes that the mitochondria is the powerhouse of the cell. That's usually the one thing people remember about it, or about them. So what that means from our standpoint, or one of the things I want you to know for this class, is that what that means is that the mitochondria use oxygen to remake ATP, to remake the energy molecule in the cell. So that's what makes them the powerhouses. You make ATP there, but you do it using oxygen. So we've got to get oxygen to them, which we use myoglobin to do. So myoglobin is a shuttle bus to do that. Now, it doesn't have to exclusively transport via myoglobin. Some of it just diffuses, but myoglobin is one way to get oxygen to those mitochondria. And then ignore the modified organelles, because normally this chapter follows the chapter 3 on generic cells. But an important term there are the myofibrils. And so these are the myofibrils, these cylinders. And so the myofibrils contain the myofilaments, which are the contractile proteins, again, that actin and myosin. And so on this drawing and every other, you're going to see that myosin is red, so that's one of the contractile proteins, and then actin is blue. And so your myofibrils, again, contain those contractile proteins, also known as the myofilaments, which are actin and myosin. And then we've got the sarcoplasmic reticulum. And so that is this sort of dark blue spider web here. That's the sarcoplasmic reticulum. And the thing I want you to know about the sarcoplasmic reticulum for now is that it acts as a reservoir or as a holder of calcium, because calcium is going to be an important initiator of the muscle contraction process, and so we store calcium in the sarcoplasmic reticulum. And then the last thing on here are the T-tubules, and the T-tubules are these kind of like purple-pink things, and so they are a continuation of the sarcolemma, which you'll hopefully remember is the cell's membrane. So they are essentially a continuation of the cell membrane inside of the cell. Because in order for a muscle cell to contract, it has to change its charge temporarily. So a resting muscle cell, the inside of it is negative, has a negative charge relative to the outside. But when a muscle is contracting or in the process of muscle contraction, you're going to get a brief period where the inside of the muscle cell switches from being negative to positive, and so that electrical signal spreads along the sarcolemma and then down the T-tubules. So the T-tubules help distribute that electrical signal, if you will. All right, so we talked about myofibrils again. So here's another depiction. So here's again an individual little muscle cell, and then you've got a myofibril here, which contains those myofilaments, the contractile proteins, actin and myosin. So what you're looking at here then, so here's the big drawing of the whole muscle cell, and now we're going to look at an individual little myofibril. All right, so when we look at it up close, you'll notice a few things. Again, we talked about striation at the outset. So striation is that striped appearance. So the cell appears a little bit lighter here, a little bit darker here, and then on a microscope it will also appear light again here. So again, the reason for that striped appearance is the overlap, which you're getting here, of actin and myosin, or the lack thereof. So the overlapped areas are darker. The areas where there's only one protein, in this case only actin, those areas are lighter. And so the functional unit of a muscle cell is called a sarcomere. And so sarcomere is runs from this little area called the Z-disc or Z-line, runs from one Z-disc to the next Z-disc. And so an individual sarcomere contains the two contractile proteins, actin and myosin, and areas where they're overlapped and areas where they're not. And so those myofibrils contain just series of sarcomeres. So you'd have one sarcomere originating over here, ending here, the next one here, the next one here. All right, and so looking at an individual sarcomere, so again, it's going to run from this Z-disc to this Z-disc, there are repeating structures. And so they run from lateral to medial in this order. So there's the Z-disc or Z-line, which is the anchoring point for your actin myofilaments. So the actin anchors into the Z-disc. And then you have what's called the I-band, which is where it's actin only. So that's this area here where you've only got this blue protein, again, actin only. And if we look over the course of two sarcomeres, as you can see down here at the bottom, the I-band extends over both sarcomeres. So you've got this sarcomere's I-band and then this sarcomere's I-band right there. So that's why the graphic looks like that. So you got Z-line, I-band, and then we've got our A-band, which is where we've got the overlap of myosin and actin. So here's your A-band on this side, here's your A-band on this side. And then we have the H-zone, which is where it's myosin only. And then we have the M-line, which is where the myosin anchors into. So from left to right on a sarcomere, it goes Z-disc or Z-line, I-band, which is actin only, A-band, which is both actin and myosin, H zone, trying to get my mouse to come back, H zone, which is myosin only, and then the M line, which is the line that the myosin anchors into. And so if we zoom in even more, so again, there's your Z disc or Z line, there's your M line. So there is your actin in blue, myosin in red, and so we'd have our I band here, A band is in through here, and then your H zone, and then your M line right there. All right, so zoomed in even more. So looking at those two in further close-up, so this is obviously a myosin filament, and one of the things you're going to see on there are your myosin heads. And so myosin heads, as you can see, have these actin binding sites. And so those two things on the myosin heads are areas where there's a really high affinity for, where it really likes actin. And when it gets the chance, myosin is going to jump up and attach to actin. But at rest, myosin can't do that because of a couple other proteins. So this is actin, again, these blue filaments here. And you can see that it kind of looks like a string of Death Stars, if you're a Star Wars fan. So you have these little indentations on actin. Those are the binding sites. And those are where the myosin heads attach. So these little indentations, again, are called active sites that myosin is going to attach to. You've got two other proteins that are important. So you've got tropomyosin here. So tropomyosin is this sort of orange thread. And so you can see that this is a muscle cell at rest. I mean, you didn't know that, but I'm telling you that. So this is a muscle cell at rest. And so tropomyosin, that little orange thread, is actually sitting on top of those active sites on actin where myosin would attach. So whenever tropomyosin is sitting on top of those active sites, then it effectively physically blocks myosin from attaching. So it separates those two proteins. So if we don't want the muscle cell contracting, we need tropomyosin covering those active sites. And then the last protein to talk about, and I'll talk about it over here where it's labeled, is troponin. And so I'm going to refer to it as troponin C because it's going to bind with calcium. And again, that's going to be really important when we talk about the steps of muscle contraction. So we've got troponin here, which it interacts with tropomyosin is another important thing to know about it, because when it changes shape, it's actually going to pull tropomyosin off of those active sites, and then myosin will be able to attach to it. So before I forget, let me show you on the anatomy app. So in terms of what we're looking at here, so this is an individual skeletal muscle fiber. So if you click on the models, go to skeletal muscle fiber, and so that's what we're looking at. So this is an individual muscle cell, and so this is its little connection to the central nervous system. This is its layer of connective tissue that wraps around the individual muscle cell. So remember that that is the endomysium. And then these little guys are capillaries. So they're going to, some of them bring in oxygen, and again, the rest of them are going to take waste products away. And then other things, so this purple thing is the nucleus. Remember, skeletal muscle cells are multinucleated. These green things, those are your mitochondria. And so again, those are the powerhouse of the cell. It's where we're going to use the oxygen. These little yellow things are ribosomes. And so ribosomes, again, are going to assemble proteins. So when we need more actin, more myosin, the little factories to do it are our little ribosomes right here. And so we can, so I can turn it a little bit. So let's look at an individual little sarcomere. All right, so in terms of some of the structures we just talked about, so you've got the sarcoplasmic reticulum. So you can see that there. And so it pops up, obviously. So again, remember, that contains calcium, which will be important for initiating the muscle contraction process. So if we hide that, and these things are the T-tubules, which again, connections to the cell membrane. So I'm going to hide those guys. All right, and so you can see all the stuff we just talked about. So here is one Z disc or Z line. Here is an actin filament. And then, so you got the I band where they're overlapping, the H zone where it's myosin only, and then here's your M line. Okay, so there's all the stuff we just found with that. And then the only other thing on this individual muscle cell model I haven't shown you is that, so again, here's your endomysium, and I'll click on it so it pops up. So there's the endomysium. And then remember that the muscle cell's membrane is called the sarcolemma, and so the muscle cells, well, there's your basal lamina. And then the sarcolemma, if I can get it to click just right, zoom in more, is that thing. All right, So you got sarcolemma, basal lamina, which is connective tissue with some extracellular fluid, with more extracellular fluid, and then we've got our little endomysium here. All right, so there's all those things. So one of the important things about actin and myosin is that they have what's referred to as a hexagonal arrangement. So now what you're looking at, and I'll actually go back to the anatomy app, sorry to toggle on you, but so now if we look at a muscle cell this way, so we look at it longitudinally, we'll zoom in more. So the view I'm getting ready to show you is looking at the muscle cell like this. So we're looking at it longitudinally. And so what you're seeing then is the larger ones here are your myosin filaments, and then these little guys are your actins. And so again, they're referred to as having a hexagonal or six-sided arrangement because each individual little myosin is surrounded by 6 actins. So here's one actin, here's the second, third, fourth, fifth, and sixth. So it's kind of a six-sided structure. And then there's overlap between them. So for example, we've got this myosin that we were just talking about, and the actin one of the actins that it attaches to, but then up here you've got another myosin. And so this myosin is also going to attach to this actin. So each individual little actin filament simultaneously interacts with 2 myosin filaments. And so again, you can see that arrangement here as well. I didn't realize I could zoom on my slideshow. Turns out I can. All right, we talked about the sarcoplasmic reticulum, talked about the T-tubules. The terminal cistern is kind of this thicker area here. I won't ask you about that in any detail yet. And so the triad is just the interaction of those 3 things. All right. So let's talk about muscle contraction, and we'll talk about muscle contraction over the course of several days, but I just wanted to touch on it now. So effectively what happens is that when the tropomyosin, I'm going to click back to it so you can see what I'm talking about. So remember that this is at rest. So normally tropomyosin is covering up these active sites on actin that myosin over here, the myosin heads, really like. But whenever we want the muscle cell to contract, what we'll do is we'll release calcium from the sarcoplasmic reticulum, which then binds to troponin C, which then causes this protein to change shape. And when this protein changes shape and sort of contracts in on itself, it's going to pull tropomyosin down or off of those binding sites. So once those binding sites are exposed, then, because myosin really likes those, as soon as it sees them, myosin is going to jump up and attach to those binding sites on actin and then pull them toward the midline. So then viewed in this closer up fashion, slide this over a little bit, go even closer, there we go. All right, so we've got our myosin head here, so it'll jump up and attach to the actin filament and then pull it toward the midline. And then these will do the same, and then this myosin head will release and it'll grab further down and pull it again, and then it'll grab further down and then pull it again. And so effectively what happens then is the actin filaments slide closer to each other. So as we pull this one in, we're simultaneously pulling this one in, and so you're pulling the Z lines closer together, and so the sarcomere is shortening. So the I bands effectively go away whenever the muscle cell shortens. So what you're looking at up here, so this is one sarcomere. So here's a Z disc or Z line right here. Here's the other Z disc or Z line. Again, the I band where it's actin only, and then A band right here where there's interaction between the 2. So whenever we've got our relaxed sarcomere here, you've got a pretty obvious I band where there's a space where there's actin only. Then when we pull actin toward the midline, which is what's happened here, the I band effectively goes away. The 2 Z discs or Z lines get closer together. So the individual little sarcomere shortens, and if we do that throughout a muscle cell, and if we do that throughout muscle cells inside of a muscle like biceps brachii, then the effect of shortening all those sarcomeres is that we're going to pull one end of the muscle, the insertion, toward the origin, and then the whole thing is going to shorten. And so that's what happens with muscle contraction, is we shorten individual little sarcomeres throughout the muscle. And so the muscle cells all get shorter, they pull one tendon toward the other, and so globally then the whole muscle gets shorter. And so that is the basics of muscle contraction. So that's called the sliding filament theory of muscle contraction. And so since we're already at 49 minutes, we'll stop there for today, and then we'll pick it up next time. We'll talk about the electrical activity that's involved in muscle contraction. So we'll see you then.